Yes. So, welcome everyone to today's episode of SwaLife podcast series. If you have ever followed drug discovery closely, you have probably noticed something puzzling. Time and again, we hear about promising preclinical results, exciting animal studies, breakthrough mechanisms. Yet, when those same ideas reach human trials, most of them fail. So, today we are asking a tough but very important question. Why do most preclinical studies fail to translate to the clinic? To help us unpack this, I am joined by Dr. Pravin Badhe, who has worked across drug discovery, translational research, and AI driven study design. So, sir, welcome.
Thank you, Shruti. This is an extremely important topic when we look, especially from a researcher point of view and for students who are still in early in their career.
Yes. So, let's begin from the basics. When people say a preclinical study failed to translate, what does that actually mean?
Okay, so that's a great start because the phrase is often misunderstood. When we say a preclinical study failed to translate, we are not saying the experiment was wrong or poorly conducted. Most preclinical studies are technically sound. What actually fail is the assumption that success in laboratory system automatically predict success in human. That's the assumption we had made with preclinical. So translational failure usually mean one of the three things which I want to mention here. The drug works in animals but shows no efficacy in patients. That may be the meaning. Second is the drug appears safe preclinically but causes unexpected toxicity in humans, or biological mechanism have collapsed when tested in real human disease. So translational failure is not a lab failure, it is a model to human mismatch. Preclinical researcher answer, does this work in this system? Where clinical researcher asks, does this meaningfully improve disease in real people? So confusing this question is very problems where the problem actually starts.
Yes, so that naturally leads us to the next question. Why do results that look so strong in preclinical models so often fall apart once we reach human trials?
Okay, so there are several uh, critical reasons and they usually interact. So first is the biological complexity. So human disease, especially the chronic diseases like cancer, neurological disorders, autoimmune disease, are not driven by single pathways. So they are network disease. But many preclinical studies simplify disease into one target, one pathway, and one dominant mechanism. So that simplification makes experiment clean, but biology is not clean. That's the first thing. The second is the species difference. Mice are incredibly useful, but they are not small humans. They are different in immunosignaling, drug metabolism, gene regulation, microbial effects. So there are a lot of things are different. The pathway that dominates disease in mouse may be buffered or compensated in humans. So that's the second thing. The third is the artificial disease model. In animal, disease is often rapidly induced, genetically uniform, and also studied in young or healthy organisms. With human, disease is slow, heterogeneous, age dependent, and the major is that it is influenced by the environment. So we end up testing drug in a system that are easier to cure than the real patient. So that's the the main issue.
So, talking about factors like target biology, disease models, or experimental design, if you had to prioritize, which of these factors contributes the most to translational failure?
Okay, so honestly, all three matters. But if we rank them, target biology is the biggest culprit. We often select target because they are upregulated in disease. they look important in pathway diagram, they are druggable. But upregulation actually doesn't mean that they are the major causing targets. So many targets are downstream effect. They are not drivers. Then they are plays important role in backup pathways. Second is the disease model. After target is the disease model where a good disease model should actually been utilized, but it actually fails of them. When we look at the models optimization process, it has got strong product strong effect, then generate publishable data, but that create false confidence. And the third is the experimental design issue, where small sample size, then short study duration, no blinding, selective endpoints. So all these experimental disease models and targets are playing important role. And target selection is the first, major uh, role player person in the, failure in the translational um, research. So this does not actually just weaken the rigorness, but it actually inflate the apparent success of the molecule or of the preclinical studies to the clinical studies.
Okay, so if we talk about oversimplification, how does that exactly distort conclusions in preclinical research?
So as mentioned, oversimplification is actually attractive because it gives clarity, but it is giving the clarity on basis of the cost. In simplified models, what happened that we are ignoring the compensatory pathways, then we are missing out with immuno and stromal interactions. Then disease progression is also compromised, and this leads to mechanistic overconfidence. So we start believing if we inhibit the target strongly enough, the disease must respond. So that's how we think. But in human biology, adapts the system reroute around the intervention. So the conclusion was not completely wrong. It was actually incomplete and overconfidently, done and has been taken further. So that's how it works.
So the next question that I have is a very practical question. What early warning signs should researchers watch out for?
Okay, so there are several red flags when we are actually working with the preclinical. The first is that the single model dependency. So if you have everything depending on maybe a single animal model or cell line, that is risky. The second is the unrealistic large effect. So biology rarely produce 90% cures. The huge effect often signal that the model has actually artifacts. Third is no dose response or time dependency. So real biology scale logically. Fourth is that no human data anchor. So even indirect evidences relevant to human genetics or patient data set or clinical biomarkers help in adding the predictability. And finally, what I think is that the mechanism that are inferred or not tested. Pathway diagrams are just hypotheses; they are not the proof. So that's how the preclinical work, and these are some of the red flag which need to be considered when you are working in the preclinical setup.
Yes. So, what tools or approaches are helping us bridge the gap between preclinical and clinical outcomes?
Okay, so this is where actually the preclinical things get exciting. First is human relevant models. So we can see that lot of 2Ds and the animals has been used in the research, but lot of human relevant models are being developed, and they are now lot of work is going on. Like organoids are there, 3D cultures is going on. Then patient-derived system where you can actually utilize the patient-derived cells and produce xenographic models. So they are they actually don't replace the animal model, but they help to add, the direct connectivity to the human system. That's the first thing that is working with the human relevant model. Second is that the system biology and network approach. So diseases are not just one target; they are networks. So drugs should be evaluated as a network modulators and not just a single switch for the target. So that's the second thing. The third is that utilizing AI and data integration. So like at SwaLife, we are utilizing this where we are using AI to integrate with genomic, transcriptomic, and clinical data. So that helps to identify the target in the real human evidences, and simultaneously it will help to develop the trace testing hypothesis before going for preclinical or costly trials. So AI is actually helping to design the experiment. So it's not replacing the complete experiment. It actually helps to focus on the important experiments and help to design the better one. So finally, if you look at translational thinking should be there from day one, so that way you can design studies by asking that what would convince a clinician and not just a reviewer or for the grant purpose. So we are not trying to convince just the reviewer. You need to think from the clinician point of view.
As we come towards the end, what advice would you give students and early career researchers who want to avoid repeating these translational failures?
Okay, so this is what I have observed is very important question here, and the podcast series which we are doing is for the purpose of early career and the student, because it is important that the student should actually first learn to think clinically, uh, even when they are working in basic research or as a in preclinical setup. You need to always ask that who is the patient and what outcome actually matters, and that is the the thinking from the clinical point of view. The second is that embrace uncertainty, where you need to welcome strong science, which actually can go wrong at the early stages before it goes to the patient and pays the price. So that is important to understand. Third is you need to understand the data, not just the experiment. So major of the students which I came across focus more on the experiment, and they are not looking at what type of data they are going to get, what stats, what data integration, and what critical thinking need to be done so that way the data can be utilized in a proper way. The fourth is that you move beyond single target thinking. So a lot of work with the cell studies or with the animal studies are focusing on the single target, but you need to learn about system biology, network pharmacology, and multi-target strategies when you are working with the leads or with the formulation. So my suggestion will be that you should ask better questions. So instead of asking, does it work? You need to ask, why would this work in human? So it is very important to that. So preclinical expertise actually open doors in industry in many areas of industries. Then academia is there, data science and translational roles are there. But if I want to give you some of the major areas where the preclinical skills can be utilized, the first is the preclinical scientist or research scientist, which is a major, direct path in industry or CROs, where pharma companies, biotech startups, and CROs are hiring. You work on target validation, you work on in vitro and in vivo studies, you are focusing on pharmacokinetics, pharmacodynamics, and toxicity screening. But the key difference which is observed today is that company just don't actually hire now to run the experiment. They want researcher, scientist who can interpret the data, understand the relevance to the disease, and the major part is that they can communicate those translational risks. So that way, the career can go faster, and the student or the early researcher can grow with this expertise. The second is the translational research scientist, which is one of the major emerging career here for preclinical people, where you are actually in between the preclinical and clinical team. So your job here is to ask, does the preclinical work, signal which got in the preclinical matters in human? The second is the what biomarker can move into the trials, and third is where is the translational risk. So if you enjoy where you can actually utilize your thinking across different system, this is the area where you should actually try. The third is target discovery and validations specialist. As we mentioned earlier, the major culprit is the target. So another strong path where you can utilize your expertise, the target discovery and validation. This role actually combine the biology, the disease mechanism, and human relevance. So it is overlapping with genomic, network biology, and system pharmacology. People who understand that why target fail are extremely valuable because they help the companies to avoid the expensive mistakes. So this is very important role, and that expertise are very important. Now the fourth one, which is coming into high demand recently, that is the computational and AI enabled preclinical role, where you are working in the intersection of preclinical biology, data science, and AI tool. A lot of our program actually doing this, where the AI tool from SwaLife, we are collecting data, and those data is then further proceed and put into predictive medicine. So when we look at the career role for these people, is one is the computational biologist, second is the system pharmacological scientist, and third they can also work as a AI assisted drug discovery analyst. So your job is not to replace the experiment here, but to make sure that you work on prioritizing experiment, predict the translational risk, and integrate human, that is the clinical, with the preclinical data. So this one is the one of the major fastest growing career. Next is the safety, toxicity, and regulatory facing preclinical role, where you will be working in toxicology scientist, then IND animal study team, regulatory facing preclinical expert. So your role requires strong experimental understanding, knowledge of regulatory expectation, and ability to design the study that the regulatory actually trusts. So the people who understand why the safety signal appear later are highly, respected in this space, and where plays important role. Then the sixth one, another major role in the career of the students or early researcher, that is clinical research, biomarkers, and early phase trial support. So again, from the preclinical to clinical, that is a middle role where you're moving into biomarker development, early phase clinical research, and translational endpoint study design. So if you understand that how preclinical mechanism work, then explore response relation and disease biology, you can actually help into designing smart first in human studies. Then one of the major one, which I have seen lot of student actually suffer from this in the early phases, that scientific writing and strategy and consulting. So preclinical experts are needed as a scientific writer who can actually communicate the result in the form of the articles, in the form of the grants. Then the drug discovery consultant role is also part of that scientific writing. Then strategy and dual diligence analyst is also major area there, and that is mainly for the startup investors and grant agency. So if you can explain the research uh, and can be easily understand. So that is major area in scientific writing. Then the last one is, the academic, or you can say academic industry hybrid career, where academics are actually collaborating with the industry. So there is a strong opportunity there where the collaborator from industry is working with the academician for translational research center, grant-driven applied research. So academician who understand the translational failure uh, can be utilized, and more funding from the industry, collaboration, impactful can be done. So all these areas are important, and students can actually utilize. Even the early career researcher can utilize them. So I will leave student with this, that the preclinical research is not a narrow career; it is a foundation skill. If you combine it with the human relevance, data literacy, and translational thinking, you can move into science, strategy, regulation, AI, or even in the leadership role. The mistake is thinking that I am only preclinical uh, worker, but in reality, you know how the idea succeed or fail before reaching to patient, and that knowledge is actually priceless. So it is very important.
That was a very powerful note to end on. Today's discussion really shows that preclinical failure isn't about bad science, it's about how we frame and interpret models. Thank you, sir, for breaking this down so clearly.
Thank you, Shruti. If we rethink how we design the preclinical studies, we can actually dramatically improve the transition from preclinical to clinical, which will ultimately help the patient faster. Thank you.
Yes. And to our listeners, if you found this episode, please share it with your colleagues and students because better translation starts with better thinking. Thank you for listening.
Thank you.